I'd like again to welcome everybody to the 197th episode of the Minnesota Outdoor Skills and Stewardship Series webinars. My name is Benji Cohen with Minnesota DNR, and today we're welcoming back Dr. Michael Joyce from the Natural Resource Institute of the University of Minnesota to talk about some research projects they're doing on fishers in Minnesota. So with that, Michael, I'll turn it over to you.
Yeah, thanks so much, Benji, and thanks to everyone for joining. Really happy to be here today to talk about fishers, one of my favorite species that we have in Minnesota. So I assume many of you, all of you, most of you know what a fisher is, and that hopefully piques your interest in joining today. If not, you can see a picture of one on the screen here. They're one of our weasels. I'll talk more about some background about them in a little bit. But they're really fun, and I don't know I have any good tips on how to see one other than to be lucky. But any day you see a fisher is a good day. So I'm going to be talking about ecology of fishers in the southern half of Minnesota. But I'm going to start sort of in the middle with some observations. And observations that sort of piqued my interest when I heard about them, piqued my curiosity. So the first one is a picture of a fisher in the Bronx. It was taken in April of 2014 by an NYPD officer. And this is really a captivating image because we think of fishers, and we have research-wise, the research community historically really found fishers mostly in wilderness. And so here's a fisher doing something that we don't expect. So the first time you see it, maybe it's a one-off, it's interesting, but maybe it's the exception. Then you see it again. And so we see this in Minnesota. Here's a picture that someone took in Hastings in 2020. I apologize. I don't know who took this picture. It was sent to the DNR. I don't have a name to give credit for. If you're watching today, thank you for submitting this. But so here's another fisher in a place that we don't expect it. And it's not really an exception anymore the more that we keep seeing this happen. And it's not just fishers, it's other wildlife, especially carnivores, that we're seeing move back into areas that they previously occupied, that humans have changed the landscape, and or as humans move and spread into areas that they currently occupy, we're having a lot more close contact. That presents some really exciting questions we can ask. It also presents some challenges of how we're going to get along and how we're going to reduce conflict and that sort of thing. And so coming back to fishers, you know, this really brings a lot of questions up. And when I saw this picture in the Bronx, these are some of the questions I had. What's going on with fishers? Why are they doing this? What's allowing them to move into these spaces? But if they're living and maybe doing well and better than we thought in areas that we don't think they could survive, maybe we need to change what we know about what they need to survive. So what do they actually need to survive? What are the minimum things that fishers need to be able to live in a landscape? But it's also important to consider that just because fishers are somewhere doesn't mean it's a very good spot for them. And so how are they actually doing in these areas that they're reoccupying compared to areas that we consider better habitat from what we've studied for the last 50 plus years on fishers in more wilderness areas? And so these are all questions that we were really interested in as we started seeing some information about fishers in southern Minnesota coming in. And I'll come back to these when I talk about the research project, which is going to be most of the talk today. But before we get there, I'd like to give more of a general background on fishers and their natural history. So fishers, as I mentioned earlier, are a member of the weasel family, or the Mustelidae. I'm a little biased, but I think the Mustelidae are one of the coolest group of animals in the world. And so they're really diverse. So we have the smallest carnivore species, the least weasel. Largest weasel we have is a sea otter. So they span a range of sizes. We have some like otters and sea otters that live in aquatic and marine environments. We have mink that are semi-aquatic, badgers that make their living by digging, martins and fishers that climb trees. So there's just a lot of diversity of where they live, how they make a living, and they're a really exciting group to sort of see this diversification of adaptations within a single group. Now, within the weasels, fishers belong to this smaller subgroup that some people call the Martes species complex. Taxonomists call it the Galaninae. It's, taxonomists do their thing. But what it is 11 species, plus or minus a few, as we're doing more work as scientists to figure out how many species there actually are, in 4 genres. So we have the Tyra, which lives in Central and South America. To me, it looks sort of like a fisher with short hair, but it looks different than a fisher too. The Wolverine, which lives sort of in this circumpolar distribution. It lives at high elevations in boreal forests, sort of across the northern hemisphere. The fisher, which we'll talk about in more detail, a lot more detail today. And then 8 different, plus or minus a few, species of martin, including our American martin or pine martin that we have here. So we have 2 members of this group in Minnesota. And so because they share an evolutionary history, even though they live in different places and sometimes do very different things, there's still a shared history, and they do share a lot of behavioral morphological adaptations. And so, you know, those folks in the science community who study one of these species are generally really interested in the others. So it's a really nice group of species, I think. Again, I'm biased to study. So just again, natural history, basic stuff. What do fishers eat? I always like to joke, but it's true. They will eat anything that they can catch and put in their mouth. They do well on snowshoe hares, and a lot of like northern Minnesota, they eat a lot of snowshoe hares. They have certain adaptations that allow them to be one of the few predators that's pretty good at killing porcupines. And so porcupines in some places are really important prey for them. And they also scavenge deer. So not necessarily off the road. This is a roadkill, but we've seen fishers in northern Minnesota that are scavenging wolf-killed deer, hunter-harvested deer. If you hunt and you leave a gut pile, they'll probably come and scavenge that as well. But they eat a lot of other things too. They eat small mammals, they eat mice, shrews, voles, squirrels. They'll eat fruit and that sort of thing too. So I'll come back to that at the end with some of the findings that we have from our study. The fisher reproductive cycle is really interesting. I always like to talk about it because they are a species that is functionally pregnant almost the entire year. And so they use what's called delayed implantation. That means that when they mate and they get an embryo, that embryo develops into a ball of cells, and then it stops and it just kind of hangs out in the uterus. And it's not until about 40 or 50 days before they'll give birth that embryo implants. And so this is really common in carnivores and some other groups. And the reason that this evolved is because it allows, reproduction is very energetically expensive. And so if you're a fisher, it's a bad prey year, it allows you to choose whether or not you're in a good condition to implant those embryos and carry them to term. For fishers, they live at low density, so it also allows them to take advantage of mating when the best time the males can find them is, but then not necessarily giving birth right away, but just waiting until the better time to give birth. And so what happens is about this time of year to mid-February, they will be implanting any embryos they have. They'll have active gestation, and then they'll give birth in March, usually. It seems like in northern Minnesota, it's late March, early April. In southern Minnesota, it's sort of early to mid-March, but there's a lot of variation. And then within about a week to 10 days of giving birth, a male, usually multiple males, will find the female at her reproductive den where she's given birth and will mate with her, and the whole thing starts again. So it's a kind of an interesting story, very different than what humans do, but maybe a similar time frame. And then in Minnesota, our litter size estimates from northern Minnesota are 2.5 with a range of 1 to 4. So it's really common for them to have 2 or 3 kits. 1 isn't that uncommon. 4 is a little less common. That's the most that they've ever been documented having, not just in Minnesota, but elsewhere. And keep those litter sizes in the back of your mind because we'll come back to them in a little bit. There we go. So fishers are, for all intents and purposes, forest dependent. And non-forest is typically not habitat. They do hunt in non-forest, they can travel through non-forest, but the resources that they need are by and large present in a forest. However, not all forests are created equal, and fishers tend to do best where we have these sort of messy forests, or what we call structurally complex forests. So forests that have large trees with cavities, living or dead, things like stumps and rocks and root masses, hollow logs, old trees and young trees in the same stand. And the reason that they're associated with these forests is because they provide this complexity provides a lot of really important functions. Cavities where fishers can use as reproductive dens, give birth to their kits, secure places where they can rest above the forest floor where predators can't get them, other features, so when they encounter predators, they can escape, thermal protection, prey habitat. And so these things tend to be really important for them. And again, one part of this critical structural complexity is big trees with cavities. We call them reproductive dens. They're where fishers give birth. So on the left is a picture in northern Minnesota of a fisher by an aspen reproductive den. On the right is a picture from the South Sierra in California of two fisher kits in one of the dens. And so these are really important. They don't have a lot of kits for their size. And so it's really important that they're able to take care of and protect the kits that they have each year. In Minnesota, 96% of our reproductive dens were in tree cavities. As the kits age and become more independent, they do occasionally use hollow logs. But when you look across North America, it's really more like 99% of all places that they take care of their kits are in tree cavities. And if they will only give birth in tree cavities. And we don't really know why that is. Martins, a related species, give birth in tree cavities. They'll also give birth in burrows and in hollow logs. But fishers don't do that. So tree cavities are really, really important resources for them. And because of this, there is some concern in Minnesota, but more broadly, that tree cavities are likely a limiting resource for fishers. And why are they limiting resource? Well, they always use among the biggest trees in the forest, which even where there's no management happening, they're rare. It's not like every tree in the forest is a big tree with a cavity. They take a long time to develop. In Minnesota, really a minimum of 80 to 95 years, it seems. And they can be lost really quickly from natural senescence, from windstorms, and during forest management. And so this is something that, we work a lot with forest managers to talk about how can we balance what we need out of the landscape and from our forest resources with the resources that fishers and other wildlife need. And I do want to point out that cavities are really important for fishers, but they're also really important for other species. And so this is an aspen tree in northern Minnesota. In 2014, a female fisher had her litter in this very opening that you see down here. We went back in 2019 as part of another study to put a temperature logger down there, and we had two northern flying squirrels pop out. And then we went back in June of that year to retrieve our temperature logger, and there was a saw-whet owl nesting in the cavity. And so I'm not trying to imply that every cavity gets used this intensively, but just, again, an example to show that the cavities that fishers need are also important for other wildlife as well. Okay, so I'm going to shift a little bit and talk about the sort of history and status of fishers in this region. And so this is sort of the pre-European settlement distribution of fishers everywhere you see blue. So covered most of Minnesota, all of Wisconsin, Michigan, into Canada, Iowa, Indiana, Illinois, Ohio. They all had substantial parts of those states where fishers used to live. They were almost completely removed from this area in the late 1800s, early 1900s due to a combination of large-scale forest conversion and forest harvest, as well as unregulated fur trapping. And so I do want to point out, and I need to update my map, so I don't always do this, we didn't lose fishers from Ontario and Manitoba. The map just needs to get colored. One day I'll get around to that. The dates you see here are the years that fishers were officially considered extirpated in each of these states. And the reason we have a question mark in Minnesota is that we almost certainly didn't lose all our fishers. Places like the Red Lake peatlands, the Boundary Waters probably still held fishers. There just weren't very many and they were officially considered extirpated. Now, as we let the forestry grow in some areas and through some reintroduction efforts, not in Minnesota, but elsewhere, fishers have recovered a lot of their range in this region. So in the UP, in most of Wisconsin, and in about the northern half of Minnesota. So in Minnesota, they recovered naturally. We never reintroduced them. We probably had some coming in from Canada. Those areas that I told you that they weren't removed, we probably had some recolonization from those areas as well. So we started to see fissures again in the 50s. We started seeing enough fissures by 1977 that they reopened a harvest season in Minnesota at that time. In Wisconsin and Michigan, there were a few different reintroduction efforts. In Michigan, they reintroduced them from a source elsewhere, and then they moved them farther east in the 80s and 90s. So this has been pretty successful. They don't go all the way down to the blue line you can see there, which is where they formerly lived, but it's not bad. But in more recent years, we have been seeing more range expansion into southwestern Wisconsin, southeastern Minnesota, out, through northwest and west central Minnesota. North Dakota now has fissures. And so again, this is where we start to see them showing up in areas like Hastings, farther south, and I'll show you the sightings in a little bit. But we also see them moving into Iowa. So since 2017, the first documented fissure back in Iowa after they were extirpated, they've had pretty regular sightings in the northeastern part of the state. Now, what's interesting is we usually get this spreading in wildlife populations where you have a population source that's doing really well, so well that not all the animals that are produced can live there, and so then they spread out. But does not appear to be the case of what's happening in Minnesota. In fact, this expansion coincides with about 50% decline in our fisher population from the late 90s, early 2000s till the 20 teens, about a 20-year period. So this is the DNR population monitoring data. The population estimate is in the dark blue, and then the number of fishers harvested is in pink. And so you can see that we've started to have our population go back up. It's not going up super fast, but that's okay. It seems to be driven by some low harvests. And so this is a concern. We've been doing some research. I'll say a few things quickly about what's likely behind this, but it is encouraging to see that they're coming back up. So the Minnesota DNR did a project led by John Erb from 2008 to 2015, where they trapped and radio collared 115 fishers. They relocated them weekly from the airplane, and it was a really comprehensive project. They collected all kinds of data on survival, reproduction, habitat use, home range size, diet. Just did a really, really thorough job. And this project wasn't necessarily aimed at understanding the decline, but with the decline, it was aimed at updating what information they had to help manage that population. And so I'm not going to go through a lot of the results of this project, but there were sort of, from that project or just from other data that DNR was collecting and has continued to collect, there's been some sign of some of the potential causes. And so overharvest is something we hear about a lot. Fishers don't reproduce fast for their size, and so overharvest is an issue and has been an issue in the past. It does not appear to be the primary driver, even though it's listed as number one. There were a few years of really high harvest that if harvest was at that level every year, it would be an issue, and it can still contribute to the decline, even if it's not at that level. But it doesn't appear to be the major thing. Instead, predation appears to be a big part of it. So bobcats in particular kill fishers, and they seem to be doing so, or at least they did during John's project, at a rate that is not sustainable for the fisher population. We also, with cavity trees and with general clear-cut logging, there are some areas where habitat loss likely played a role in that as well. And so I'm happy to answer questions about this at the end, but I'm not going to say a lot more about this now. So moving on to the range expansion, the first confirmed record of a fissure in southern Minnesota, south of I-94, was in 2005. And so John Erb and the DNR started collecting these. They get one or two a year, some years none, until about 2013-14, where things kind of upticked, and then it really took off from there. And another slide I need to update. This number from 2020 really should be 23. So it's a lot higher than that, what that line indicates. And so this is really good information to know that they're moving down there. And it's tempting to look at this graph and this increase and say, wow, the population has really expanded. The problem is we sort of get these public sightings, most of them, when people are interested in sharing. And part of the increase is more people having trail cameras, more people knowing that we're interested in these sightings. But it still is compelling information that we have fishers and that in some areas they seem to be doing pretty well. So this is a map that's outdated. It's from 2020 when I made the last graph, but you can see the pink dots are some of those sightings. The green in the background is forest cover. And so what you can see is a lot of these sightings that we're getting are in areas with forest cover. That's what we would expect. So especially along the Minnesota and Mississippi rivers, we've got a lot of forest cover down in the southeastern part of the state, the driftless zone. There's some good chunks of habitat, and so we see them there. But we also see them in areas that don't have a lot of forest cover, like near Hayfield, Minnesota, and some of these sightings as they're moving through this landscape between forested patches. And in the Twin Cities metro, where there is forest, but there's also a large human impact on that landscape. But we know they're reproducing in some of these areas. Here's a picture from the metro of a female with one of her kits from that year. We've seen this elsewhere. People who've shared trail cameras of fishers with their kits in southeastern Minnesota. We also see them again in these areas where we don't think of as fisher habitat, agriculturally dominated landscapes. And yeah, you can see there's trees, shadows of trees in this, but that's not necessarily where we expect them to show up and to live. And so we, with Environmental Natural Resource Trust Fund support, we started a project on the Southern Minnesota Fisher Project in 2022. It'll continue through this June. And our goal was to determine the distribution status and general ecology of fishers in Southern Minnesota. First, to learn about them, because it's an interesting situation. I had all those questions that I started this presentation with. But second, to provide information that managers need to decide how to manage these populations. And so distribution status, it's been really nice to get all these public sightings, but there's a lot we don't know when we know, well, there's a fisher there on this date, but then what happened? And so that's sort of the next step we wanted to take with this project. And as you can imagine, there's a lot of differences between northern and southern Minnesota. So where our core fisher population has been since it recovered through the 50s in 1977 has been in the Laurentian mixed forest province, and they've moved into this eastern broadleaf ecological province. They're not the same, so what does that mean for fishers? And so a big part of the project is to collect this data so we can compare and see how they're doing in northern and southern Minnesota. And so we have a lot of different questions and comparisons we want to make. Distribution and population status, especially in the metro. Where are fishers living and reproducing in residence versus where are they willing to move through? A population size, how many fishers can we have in southern Minnesota? Home range size and density. So a home range is how much area an animal needs to meet its needs and to live and to survive and to reproduce. And home range size has a strong impact on density, especially for fishers because they're territorial. So what does that look like in southern Minnesota? Is the habitat worse and do they live at lower density, or is it better and they live at higher density? A diet, what are they eating? That's really important to understand how the population is doing. Food is such a critical part of how an animal makes its living on the landscape. But also, as a predator moves back into an area, are they having impacts on some of their prey? We don't know if we don't know what they're eating. Reproduction litter size, again, a really good estimate of how well they're doing. Are they living there, but they're not reproducing as frequently, or they have smaller litter sizes? That would tell us something different from if fishers in southern Minnesota were reproducing, for example, better than fishers in the north. And survival and causes mortality. So what we've seen with a lot of urban, suburban carnivores is that there's high risk, high reward. Good resources, that's what brings them there, but a lot higher risk of mortality because humans drive cars and don't always tolerate them and things like that. So those are the questions we set out to answer, at least get preliminary data on. And I'll kind of go through the rest of the talk and give you some preliminary data and talk a little bit about how we're answering these questions. And so with the project, there's two main parts. The first is a camera survey. The camera survey is really aimed at understanding the distribution and population status. And again, these public sightings are really, really helpful, but we need to go out and do a systematic camera survey so that we know what that means. Getting a picture of a fisher once means something different than if you get that fisher on camera numerous times. That gives us more evidence that, yeah, it's probably living there. It's not just passing through. And so what we did is we divided and found 2 study areas, one covering a lot of the parks in the Twin Cities Metro, another one down in southeastern Minnesota. We put out for 2 years, 100 cameras a year, roughly, 50 in the Metro, 50 in southeastern Minnesota. And at each of the sites, we put out a single camera and we left them out for about 6 weeks in the fall. And then we got them, went through the images and analyzed where we're getting fissures. So I get a lot of questions on how you can detect fissures on cameras. And so I always like to address this. This is the design we used. And so what we would do at each site is we would find microhabitat. In this case, a log. It may be a rock outcrop, a stump, the base of a large tree that has a cavity in it, or a squirrel nest or something like that. So some microhabitat that a fisher might be interested in anyway. We would aim a camera at that microhabitat, and then we put 2 ounces of salmon oil right where the camera was aimed. We'd put some skunky smelling lure up in a tree that'll get them to come in from farther away. And that seems to be really effective, not just for fishers, but for many, many other carnivores, and squirrels and deer sometimes come and check them out as well. And we didn't know what to expect. We knew there were fishers. We didn't know how many. We didn't know where they'd be, but we found quite a few fishers, and that was really exciting. So here's just, I'm going to go through some of our images. Here's a fisher checking out our salmon oil. That sort of dark stripe you can see is the salmon oil. One of the reasons we like to use that is it can rain, and this scent's going to be there for 6 weeks. Here's another fisher, female fisher, checking out one of our sites, smelling the salmon oil. Here's another one. And I could go on like this for a while. I won't, although it is fun to look at fisher pictures. But what did we find over 2 years? And we are still analyzing some of these data, but generally what we found is that in the Twin Cities Metro, we have breeding residents every year in some areas. But there seems to be more turnover and definitely fewer fishers because there's not as much habitat available. And so we might have a female that is in one of these areas. She breeds for 3-4 years. She moves or she dies. And then that area won't necessarily be reoccupied, maybe for a few years. To contrast that, in north of the Metro in the Cedar Creek Ecosystem Science Reserve area and down in southeastern Minnesota, we have consistent resident populations where it seems that all of the suitable habitat is very well occupied. So if one fisher dies, another one's going to probably move in and take that available space. And so this is kind of what we expected we might find, but it's really good to have data that helps us understand this, because as you look at the larger region, we might want to consider managing fishers a little differently in the Metro than we would where there's more contiguous, consistent resident population. The second main component of the project involves live trapping and GPS collaring fishers. And so we put out live traps. Here's some pictures of some of our sets. We use these cage traps. We like to put leaves and stuff on the bottom to keep it so that they don't have to stand right on the trap. We put these white or sometimes brown tarps over them. That protects the fisher in the trap. So if it's windy, if it's snowing or rainy, they're not going to get wet. And then we put natural vegetation to add more insulation and to make them look more natural. And so we check these at least once a day. If we are trapping in the Metro right now, if you encounter our traps, you should know, don't touch them. It is actually illegal to disturb them. We're permitted. But I also like to make the point that all of the things we're doing with live fishers is approved and permitted and follows a pretty stringent protocol. And we are primarily concerned with their safety, health, and well-being, and secondarily interested in our research objectives. And I just really want to make that point, because if we're hurting individuals to study them, then we shouldn't be doing what we're doing. When we catch them, we anesthetize them, we take samples, we put on collars and ear tags, we monitor their health, and then we let them go. And I'm going to show this video twice. I never get sick of seeing it, but this is a slow motion video of a fisher we're releasing after we've processed her, and you can see her bounding away. So now I lost my mouse. There we go. So I'll play this a second time too, in case you miss it. So really good example of how they move. And I'll play it one more time here for my benefit, if not for yours. And then, so it's fun to trap them. It's fun to see them up close and handle them. But really, for me, the fun part is when we let them go and they go back to their normal life and we can follow them and see what are they doing and why are they doing it. This is an example from Cedar Creek of two fishers, the location data we got. Neither of these fishers is actively collared right now. One of them died earlier this year. The other one, we removed our collar from last year and just let her go. But what you can see is each of these points is a fix that our GPS collar got. And so we get really, really detailed information about where they go. And one of the cool things, I said they're forest dependent, but they can sometimes move through open spaces. If you look close at these images, you can kind of see that I'm not making that up. So here's an open area, and even when they're near open areas, they tend to hug the forest or some cover, even if it's a shrubby area. But this is really good information. We can see how much space do these fishers need? Where are they going? Where are they not going? When are they moving? So the collars give us really rich data to answer a lot of those questions. Here's another fun example. It doesn't always go how you think. So we collared this male. We didn't expect him to disperse, but he had other plans. And so we collared him here in early December. And then for about 3, 4, 5 days, he made his way about 6, 7 miles up here where he lived for a few months. Something changed for him. I don't know what, but in late February that year, he decided, yeah, I don't really want to stay here. And then he again made another 7, 8 mile move and set up shop in another area. Unfortunately, he was hit on the road by a vehicle. So we actually lost track of him, got the collar back, found out what he'd been up to. But, really kind of interesting. And we can see what path he took, when he moved, what time of day, all that information that can help us better understand how they are navigating this fragmented landscape. So just a quick summary of our GPS telemetry. Our goal is 28 collars. We have deployed 27. Yes, as of yesterday. We have 2 more collars to go because we got one of them back early enough that we can redeploy it. The collars are getting a location attempt every 1 to 3 hours. As you can imagine, if they're in a cavity, we don't get a location then. But there's a radio on it, so we can go listen, see where they are, download the data remotely. It's got an activity logger. I'll show you that data in a little bit. And these collars last about 8 to 10 months, sometimes 11 months. So we're getting a lot of rich data for a good chunk of these fishers' lives. Not lives, but years, the year that we're studying them. So from those locations, I won't go into details. We use a variety of statistical models to estimate home range size. And one of the things we did not expect to see is that female fisher home ranges in southern Minnesota are a lot smaller than they are in northern Minnesota. So 6 square kilometers on average in the south, 16 in the north. And to put that into context, this amount of space that a female fisher in southern Minnesota uses is very similar to what a pine marten female in northern Minnesota is using. So it's not a lot of space. That means we can potentially, where there's habitat, have higher density of fishers than we do in the north. Males, we have not been able to collar as many males, and they've been a lot better at breaking their collars off. And so we do see a similar size, but maybe a little smaller for males in southern Minnesota too. They just tend to be a little bit more similar. So about 36 square kilometers in northern Minnesota. And how are they able to use such smaller space? We think it has to do with diet and prey availability. And so the way we're studying diet is we're using good old-fashioned scat analysis. So we're collecting what we like to call brown gold when we're out in the field tracking them. We'll collect the scats, we'll take them back to the lab. We'll literally wash them through a sieve, take all the undigested prey remains, so things like berry seeds and skins, bones, hair, teeth. We'll identify those, and then we'll have a good idea of what they were eating. We're also using a process called stable isotope analysis, and it basically is built on the principle that you are what you eat. And so we can take a chemical signature from their hair, we can compare that signature to the signature of the different things that they might eat, and then we can get a relative idea of what they're eating more than other things. And so by combining these methods, we're getting a lot more complete data on what they're eating. So what are they eating? You can see some comparisons here. This is preliminary data. We have about 150 scats we need to go through in the next month or two. But what we see in some cases is they're eating similar things, but the species differs because of where they live. So in northern Minnesota, they're eating a lot of snowshoe hares. Where they're eating rabbits in southern Minnesota, it's cottontail rabbits. But there are some other differences. So like in the north, we're seeing porcupines, jumping mice, redback voles, deer mice is what peromyscus are, shrews and moles. And in the south, we're seeing a lot of squirrels. We are seeing some mice and some voles, but we're seeing a lot of gray squirrels, a lot of chipmunks, a lot of flying squirrels. And that does seem to be something that is different in prey availability. There seem to be a lot more squirrels down in where we're studying them in southern Minnesota, which is possibly to likely why they have smaller home range sizes. We see them eating possums in southern Minnesota. We have not documented that in the north. There are a few other things that this will probably change as we go through more samples, but grouse, waterfowl, snakes, insects, scavenging deer we've actually seen in southern Minnesota now. We've seen fish in southern Minnesota. And then plants, fruits. In northern Minnesota, they're eating choke cherries and sorbus or mountain ash. In southern Minnesota, they're eating a lot of black cherries and crab apples. So again, this is not final data, but it does give you a sense for what they're eating and how they're making a living. The activity. So we are interested in activity patterns as well. As I mentioned, the collars basically like a smartwatch. They have an accelerometer that tells us when they're moving and not. And so this graph, this is a longer term graph, and then here's kind of a zoom in. We're getting a reading every 90 seconds, and basically the larger the number, the more energy that fisher was using as it was moving during that period. So it's really easy to see here periods of inactivity followed by periods of activity. And in some cases, these are really high values. This fisher was probably moving pretty fast, maybe encountered a predator or another threat, and that's what made it do that. So these data take a long time to dig through, but we're really interested in how much variation is there between individuals and when they're active, and how do things like temperature, time of year, what they're eating, how does that influence when they're active? And so that's something we'll continue to dig into. And getting towards the end here, but reproductive dens, litter size. So the collar emits a radio signal we can listen to, and then we can go out and find the tree cavities where they're giving birth and caring for their kids. And so it's really obvious when they have litter, because rather than moving throughout their whole territory, they settle down and they're at the same tree day after day. And so here's some examples of some of the trees, big trees, cavities. We found a lot of oaks, some aspen. And here is the first litter we documented. And so we put up cameras at these dens. It's hard to see here, but this female has a kit in her mouth. So here's her first kit. Here's her moving her second kit, looking around. So we have 3 cameras, that way we don't miss them. So she's looking around, making sure it's safe. Here she goes with kit number 3, and here she is with kit number 4. So our first litter, and this female, we've tracked her 2 years in a row. She had a litter of 4 both years. So we've found, these numbers are probably a little out of date, but somewhere around 30 to 35 reproductive dens in southern Minnesota. And when you look at our current litter size data, very similar to northern Minnesota. So average of about 2.5, range of 1 to 4. We'll hopefully add another 5 or 6 litters this year while we're tracking. But really good information. And again, they're living in smaller space and reproducing just as well. That indicates that they're not just down here, they're doing pretty well. Survival causes mortality. We're still digging into this. This is something that we're going to be looking for a second round of funding to better understand survival, because you need more sample size to understand this. But preliminarily, we are concerned at the rate of roadkill mortality. It is much higher than what we've seen in northern Minnesota. We've had some predation, seems to be coyotes, but it seems like maybe that's lower than northern Minnesota. So maybe those things balance out. But we do have some concerns about health and diseases. We've documented fishers in southern Minnesota that were exposed to anticoagulant rodenticides. We have some individuals that we're concerned may have died because of health and diseases, and we're doing some surveillance on that to try to figure that out. So this is very much an open question, but they're using not a lot of space, they're reproducing well, but if they're not surviving as well, then we really need to know this to know really how well they're doing. So that is the presentation. I am not going to be able to list everyone by name, but I'm here talking. This is the result of a ton of people working collaboratively on this project, including members of my lab. I will mention Adam Mortensen is a grad student studying this. A lot of folks, Three Rivers, Park District, Dakota County, Minnesota Zoo, Cedar Creek, Friends of Mississippi, Minnesota DNR, some other folks. And then I do need to mention again that this project was funded by the Minnesota Environmental Natural Resource Trust Fund. We've really appreciated their support for a lot of the research I've done over the years. And with that, I would gladly take any questions.
Nice job. It sounds like a super interesting project. And we were talking before this, you get to get out and did you collar one in Hastings yesterday?
Yeah, in that area we did. Yep.
Nice.
It's always fun for me to get out of the office.
And you come pick a better day for it. We had a great audience today. I appreciate everybody joining us on it's a balmy January day today. So
yeah,
we'll take it. So we had a couple of questions in here before I saw one of them in here. I just lost it now, but one of them was on nesting boxes for fisher. And I know you published a paper on that. I can put that link in the chat, but I didn't know if you had a better link for that.
Yeah. So right now we haven't done any nest box work in southern Minnesota. We figure we needed to answer these more basic questions first. It does appear that where they are living in southern Minnesota, there seem to be more cavities. We haven't actually done a measurement on that. We'd like to. But where people have public land and are interested in seeing them, you can get in touch with me by email and I will send you our building plans for that. That was another Natural Resource Trust Fund project that we'd be more than happy to share that. And it is a fun way to see fishers and other things, especially squirrels, that like to use those boxes. So if anyone's interested, please follow up and I'm more than happy to share information on that project as well as the building plans.
Sounds good. So Mark, if you're still listening, you do that. I did put a link to the paper in the chat too. That probably has your email address also. So
perfect.
I'll let you take your presentation down and people copy that down also. And I was just going to say, John put a nice New York Times headline in there. The Chicago police pulled a live coyote from the supermarket just last week. So it's closer than we anticipate sometimes.
Yep.
Barry was asking, I kind of get a kick out of this one. What would you include in the skunk junk you put in the tree? What kind of recipes are you using to attract them in?
Yeah, so skunk junk is actually a branded product. It's a type of trapping lure you can go and buy. I don't know what's all in it, but it's at least made up of skunk essence, so their gland secretions, and Vaseline. So it doesn't freeze. You don't want to get it on your clothes unless you want to throw away your clothes. I lost my favorite field shirt by accidentally rubbing into it. But there's other things like Gusto and you can go to Minnesota Trapline or a lot of different trapper supplies and purchase skunk-based long-distance call lures. That's just the one that we use to keep everything consistent. Good question.
Interesting. Adrian was wondering, he says, thank you. Was wondering about identifying fisher predation on the skulls of their prey. He read somewhere that a well-fed fisher will just eat the brains out and leave the rest of it. He was wondering if you've seen anything like that in your research.
So weasels in general can do that. What I'll say is that where they don't consume most of what they kill, it's generally because it's a deer that they're scavenging or something else big that they're scavenging. With most of the prey they consume, they take the whole thing. And if they can't eat it at once, they'll eat it later. So like at the reproductive dens, we see them bringing a lot of squirrels back. I know there was one, we went to swap out our SD cards and our cameras, and there was like 3 dead chipmunks, 2 dead mice, and a gray squirrel at the base of this tree. So they have to sort of, they can't assume that because prey is available today, it's going to be available tomorrow. And so they will eat anything they can carry, they will eat all of it, either in the same meal or they will try to cache it somewhere that they can.
Interesting. Paul was wondering, when he's deer hunting in northern Minnesota or northwest Wisconsin, is he more likely seeing Fisher or Martin?
Well, it's going to depend on whether you're in Minnesota or Wisconsin and where exactly you are. It could be either. I regret now not adding a slide for comparison. So if you see an animal that's more the length of a gray squirrel with the tail, you're probably seeing a marten. And if you're seeing something that looks a lot bigger than that, you're probably seeing a fisher. Now, they're not actually as big as they look. Both of the species have a lot of fur. So, like, we get some concerns from folks about fishers eating cats. The average domestic cat is substantially bigger than the average adult female fisher. They're only about 6 pounds. Male fishers can get up to 14 and a half, 15. They can get a little bigger than that. They don't often. But yeah, so using the relative size, if it looks like it's in the size of like a gray squirrel or a fox squirrel, it's probably a marten. That would be a really good indicator if you're sitting in a deer stand.
Interesting. Fran had kind of a question similar to that. Can you compare a Fisher and a Martin tracks in snow?
So it's because they're different in size. So martens are really only, male martens are about 1.1, 1.2 not kilometers, kilograms. So they're about 2.5 pounds. Because they're so much smaller, their tracks are different size. And so there are a few papers that have been published that if you take the right track measurements, you can have pretty good confidence of whether it's a fisher or a marten. And like male fisher tracks, they're so much bigger than a marten. It's pretty obvious. There's just a little bit of size overlap between a female fisher and a male marten. Same with female martens, are pretty obviously smaller. But they tend to have the sort of 2x2 lope pattern pretty commonly.
Okay. Mary is wondering, how likely is it that fishers will return to the same nest location or the same location to nest again? Yes.
Now that depends on what you mean by location. So the same stand, they can. They don't typically use the same cavity tree in multiple years. Although they do some places they've been documenting doing that, especially if there's not a lot of options. But we think that's probably for a number of reasons. One is they also move, they use about 3 different cavity trees to 5 different cavity trees to raise a litter. And so they move if a predator finds them, they move if it gets really, gross from their kits defecating and urinating in their nest parasites. But we've had a lot of fishers in subsequent years that have been very close, but have been in a different tree cavity. And they do tend to like a forest stand that has multiple tree cavities.
Interesting. Gary is wondering, this is a good question. A year ago, he was out with his dog and his dog treed a fisher. Do you want us to report sightings of this? And if so, how and where?
Yeah, so shoot, I should have had an email about that. So or I should have had a slide about that. I'm sorry. You can email me. I would be interested in any sightings, especially we like them with images just so we can help confirm. But if you have a sighting that you don't have an image, I would love to have you share that with me. My email address was on the last slide, but it's joyc0073@d.umn.edu. I'd be happy to hear about it.
Oh yeah, we'll throw that in the chat here for you too. So, see, Mary was asking, I'm interested to know where your cameras are in southern Minnesota. I saw a fisher a couple years ago one winter in the Carleton College campus. It was on the ground and being chased by a bunch of crows. This sighting was in the winter after there was a sighting on a trail camera.
Yeah, so and I've talked with some of the folks at Carleton County about some of those sightings. So we're farther down on state forest land and WMA land, like Whitewater is where we did that study. Whitewater WMA had a lot of our cameras. So down closer to Winona is where our southeastern study area was. If we had enough cameras and time, we would love to cover the whole area, but at some point we're just spending extra time and we don't need that much.
Yeah. I had a couple of questions on the collars. One, how do you get the collars back? Do you trap them and then remove the collars? And then somebody's asking about collars with longer battery life that allow you to research them longer.
Yeah, so when we choose a collar, there's a lot of consideration given to the animal. Fishers are moving in and out of cavities and tight spaces. So we have standards of body weight we won't put a collar on that's more than 5% of the body weight. But the collars that exist in the 5% for female fishers are just too bulky and would get in the way of them going in and out of places. And so until someone develops better battery technology, I'm not willing to do it. I'd rather have the collar die early. And it's not like collaring doesn't interfere with their life, but we want it to have the minimum impact we can. So that's one of the things is we could go higher weight wise, but we don't want to get in the way of what they need to do. How do we get them back? Sometimes they remove them for us, which never makes us happy, especially when it's a month after we put them on. But we do try to trap and retrap and remove them when we can. It's not always possible because they don't always like going back into traps. And it can be really hard to get enough funding to ensure that we can do that. But we do that whenever we can.
Lisa was asking, are they more active at night or mostly during the day?
So yes is the answer. They're active when they're what we call cathemeral. They're not nocturnal, they're not diurnal, they're not crepuscular. They can be active any time of the day. And between populations and even within populations, some individuals tend to be nocturnal, some tend to be diurnal, some switch seasonally, and some just are active whenever they want to be. And so what we've seen in southeastern Minnesota is a lot of our females, when they give birth, are primarily active at night for the first few weeks after they give birth. That makes it hard if you want to stick a video camera down the den and get a litter count because she's always there when you're trying to check on her. But so it varies. It's like humans. We have some people that are night people, night owls, some people that really like to get up and get going, some people that switch depending on, in the summer, I might be stay up later with the sun. So there's no standard strategy. They do whatever they want.
Sounds like my teenagers.
Yeah, they're basically just teenagers with fur.
Kelsey has a question, and I might put a link to a previous talk you did on here, but can you expound upon what you've learned about cavity nesting animals through your research on fishers and how it relates to forest management?
Well, first of all, I think there's a lot of good opportunities for wildlife and forestry people to work together to make sure that it's not a cavity nesters or forest products. It's got to be both. And that's, at NRI especially, we focus a lot on balance and not too far one direction or the other. So because the cavities take a long time to develop, if you rotate aspen at 60 years, you're not going to let them redevelop. And so it's a simple matter of how can we work together so that we have retained trees that escape the harvest. And there's really a lot of opportunity there. And we've had some really productive conversations with folks because, again, forestry doesn't want to disrupt what other things need the forest. We all want to do things in the way that promotes what we need, right? But other things we've learned, some of the things I think have been really interesting is, with some of our flying squirrel work and some of our martin work and fisher work and that, we see fishers and martins and their prey using different cavities in the same tree at the same time. I wouldn't think that would be a good strategy if you're a red squirrel or flying squirrel, but we see it happen. So that's one of the interesting things we've seen. Or like a fisher's gone and there's red squirrels climbing up and down the tree that has its kits. And then 2 days later, you see a fisher carrying a red squirrel up to the den. But it's really interesting. These shared patterns of use don't always go as we'd expect. We see a lot of it, and we saw this with our den box project where a fisher's there, and then when it leaves, a squirrel will come in. That's more what we would expect, but we do see some co-use in ways that was kind of surprising to me. I hope that
Interesting.
answered the question.
I think so. It's an interesting topic too, and I know Elena West was talking about red-headed woodpeckers and kind of how they use tree cavities and stuff too. And there's a lot of, I think we had the same question during that episode. So,
And they've
it's,
actually seen some fishers visit some of their cavities and try unsuccessfully because the hole's too small to get in, which is really interesting as well.
yeah. Carol had one other question on do you recognize any population correlation with changes in snowshoe hare fluctuations in northern Minnesota?
So that's the one reason why it pays to be a generalist like fishers. I didn't use that word, but they can eat a lot of different things. And so when the hare populations are less abundant or small mammals, they can just eat something else. It doesn't usually happen that all prey are at the low point at the same time, but when it does happen, and I'm not familiar with any studies on fishers, there's one from Ontario on martens. It can be bad. So, like in Canada, the lynx, when the hare population crashes, they move great distances looking for hare. And they saw the same thing with marten. So, if you did get a synchronous decline of all prey at the same time, we could see a similar thing with fishers. I'm not aware if that's happened. Or if they're living in a rural area, maybe they would turn towards garbage, or I'm speculating now, but yeah, so it usually isn't that they're all unavailable at the same time. So they can do their best to find something else usually.
Great. Again, like my teenagers, they will whatever you give them, right? So,
And like me.
Michael, I can't thank you enough for joining us today. I know we got several questions in there relating to coons and some other things in there, and I don't have time to get to all of them. But I think you did a pretty good job of answering most of them in your presentation. So we'll put a link to that, or the recording will be linked on our website, mndnr.gov/discover too, so you can check that out if you missed part of it. Want to remind everybody next week we're talking to Luke Adam. He's a teacher, does a lot of outdoor classrooms. And I don't know about you, Michael, but I would have loved to be able to do some outdoor skills thing, outdoor classroom stuff, and get into this career a lot younger. So it's really
That
cool.
was summer for us.
Yeah. It's kind of a cool topic. So next week we're going to be talking
cool.
to him about outdoor classes in school. So thanks everybody. Enjoy the little warm spell we have here in late January in Minnesota, and get outside and be safe and have fun. We'll talk to everybody soon.